advanced concept presentation of the biochemistry of adenosine triphosphate. And we've been asked to take glucose and kind of generate ATP, synthesize it, and then ultimately regenerate it. So this is a quick overview of that process. We're going to go through the glycolytic and pentose phosphate pathways and ultimately through the de novo synthesis, nucleotide biosynthesis of AMP, which requires intermediates from the citric acid cycle and amino acid synthesis, and ultimately look at cellular respiration to regenerate the ATP. So from glycolysis, we could start with our glucose molecule. Now it might be tempting to put it in the glycolytic pathway and stop at fructose 6-phosphate, but I think it's more efficient if we generate GAP molecules, because if we take the GAP molecules into the pentose phosphate pathway and use mode 1, we can actually get, I think, better yield for 5-glucose 6-phosphates. And with this pathway, we get 6 ribose 5-phosphates, which is really the goal of this. So we take our fructose 6-phosphate and our GAP molecules and generate ribose 5-phosphate. I think we actually have CO2 as a byproduct here. I know that we were given that as a starting material, but I don't think we need it. I think we just use this. But next step would be to take the ribose 5-phosphate, convert it to PRPP, and use it in purine synthesis, which is a complex pathway with 9 intermediates, which you can see here that we've drawn out. But I think something that stands out is that it requires several amino acids, THF and bicarbonate, before we can take our PRPP and generate inosate, which is going to be the precursor to our nucleotide. Now, to be thorough, I think we should take a step back and look at where we're going to generate these amino acids and other components that we need. So you see here, I took our little section from the amino acid synthesis to show how we make glycine, aspartate, and glutamine. Aspartate and glutamine come from citric acid cycle intermediates, oxaloacetate and alpha-ketoglutarate, respectively. And glycine comes from 3-phosphoglycerate, which is a glycolysis intermediate. THF, if we took our PRPP and went through the pyrimidine synthesis pathway, it would combine with arididate and ultimately become orotate or something like that, and become urididate, UMP. And if UMP became deoxy-UMP, it could be converted back to TMP, which would have THF as a byproduct. So that's where we could generate our THF from our glucose molecule, and then ultimately the bicarbonate is going to come from our carbon dioxide, just converted to bicarbonate by carbonic anhydrase in the blood. Now, back to the nucleotide synthesis, we could take our IMP inosate molecule and go through this pathway to get AMP, which we're also going to need more aspartate, which we've already covered, and we're going to generate, have fumarate as a byproduct, which we also had another molecule, fumarate, generating IMP. So we could shuttle those back to the citric acid cycle. But ultimately, the point of generating AMP is to either go on to create deoxy ATP for DNA replication or synthesis, and or ADP, which could be used in cellular respiration. Now let's go back to the citric acid cycle and look at what we used there: oxaloacetate and alpha-ketoglutarate to generate the amino acids we mentioned before, and then fumarate comes back. And then finally, if we want to look at how we regenerate ATP, we can go all the way down the cellular respiration path, which is glycolysis to oxidative decarboxylation of pyruvate, which would be converted to acetyl-CoA, put in the citric acid cycle, which would generate the NADH and FADH2 we would need to drive the electron transport chain. Oh. Or we could take our, the other way we could regenerate ATP through this process would be to just shuttle our AMP to ADP and then have it down available for conversion to ATP. And finally, just a summary now that I'll take a step back and we can look at the whole board here and I can show you what we went over, which is glycolysis, so glucose in through the glycolytic pathway to get the GAP molecule, which is sent to the pentose phosphate pathway, ultimately to generate ribose 5-phosphate, which is converted to PRP, which is used in purine and pyrimidine synthesis, but in this case we're going to send it through the purine pathway and ultimately to the pathway to convert it to AMP. This would require amino acids, which would be generated from the citric acid cycle and would have fumarate as a byproduct, which would go back to the replenish the citric acid cycle, and ultimately our AMP could either be shuttled back into cellular respiration, or we could drive our glucose down to power it. And that's it. Hopefully I was closer to 5 minutes this time. It's been a pleasure spending a blistering 8 weeks with everybody, and I wish everyone the best.